Chapter 5 Membranes. Membrane structure. Cellular membranes have four components. First, phospholipid bilayer. Second, transmembrane protein. Third, interior protein network. 4. Cell surface markers. It's both transmission electron microscope and scanning used to study membranes. Freeze fracture visualized inside of membrane. A cell frozen in medium is cracked with a knife blade. Then the cell often fractures through the interior hydrophobic areas of the lipid bilayer, splitting the plasma membrane into two layers. The plasma membrane separates such that protein and other embedded membrane structure remain within one or the other layer of the membrane. The exposed membrane is coated with platinum, which forms a replica of the membrane. The underlying membrane is dissolved away and the replica is then viewed with electron microscopy. Phospholipid structure consists of glycerol, 3 carbons, alcohol. 2 fatty acids attached to the glycerol. It's nonpolar and hydrophobic. Phosphate group attached to the glycerol, polar and hydrophilic. Spontaneously form a bilayer. Fatty acids are on the inside. Phosphate groups are on both surfaces. This is a phospholipid bilayer. The head is polar and the tail is nonpolar. The head is consists of the red one is glycerol and the yellow one is phosphate group. The tail consists of fatty acid. Membrane structure. Environmental influence on fluidity. Saturated fatty acid makes membrane fluid than unsaturated fatty acid. Kinks introduced by the double bond. Most membranes also contain chloride. Stereo warm temps make membrane more fluid than cold. Cold tolerance in bacteria due to unsaturated fatty acid. Membrane protein. There are various functions. Number 1, transporter. Number 2, enzyme. Number 3, cell surface receptor. Number 4, cell surface identity markers. 5, cell-to-cell adhesion protein, and 6, attachment to the cytoskeleton. So this is the picture of each function. Diverse functions arise from the diverse structure of membrane proteins. Peripheral proteins, untouching molecules attach membrane proteins to surface. Heteromembrane protein span the lipid bilayer. Transmembrane protein, nonpolar region of protein within interior of bilayer. Polar region of the protein protrude from both sides of the bilayer. Transmembrane domain, span of the lipid bilayer. Hydrophobic amino acid arranged in a helix. Protein needs only a single transmembrane domain to be attached in the membrane, but they often have more than one such domain. Pores. Pore is an extensive nonpolar region within a transmembrane protein. It can create a pore through the membrane. Cylinder of B-sheet in the protein secondary structure. Interior is polar and allow water and small polar molecules to pass through the membrane. So this is a picture of pores. Passive transport. Passive transport have three types. First, diffusion, second, facilitated diffusion, and three, osmosis. Passive transportation do not require energy. It goes with the concentration gradients. Diffusion is a movement of molecules from high concentration to low concentration, and it will continue until the concentration is at the same in a region. Membrane is selectively permeable. Major barrier to causing a biological membrane is the hydrophobic interior that repel polar molecules but not nonpolar molecules. Nonpolar molecules will move until the concentration is equal on both sides. Limit permeability to small polar molecules. Very limit permeability to larger polar molecules and ions. Second, facilitated facilitated diffusion. Molecules that cannot cross membrane easily may move through proteins. It moves from higher to lower concentration. Passive, no energy. And channel protein. Hydrophilic channel will when open, for example, ion channel. Carrier protein bind specifically to molecules they acid. Channel protein. Ion channel allow the passage of ions. Gated channel open or close in response to acid stimulus, chemicals or electrical. It have three conditions determine direction. First, relative concentration on either side of membrane. Second, voltage difference across membrane. And third, gated channel open or close. This is a picture of channel proteins. Carrier proteins. It's help transport ions and some sugar and amino acid. Required a concentration different across the membrane. It must bind to the molecules they transport. Saturation rate of transport limit by number of transporter. For example, think like people exiting a classroom through only 2 doors versus 1 door. This is a picture of carrier proteins. Third, osmosis. Cytoplasm of the cell is an aqueous solution. Water is solvent. Dissolved substances are solute. Osmosis is net diffusion of water across a membrane toward a higher solute concentration. Water moves from pure to less pure, like this. Osmotic concentration. When two solutions have different osmotic concentration. It has three types: the hypotonic solution, hypotonic I, hypotonic solution, and isotonic solution. The hypertonic solution has a higher solute concentration. The hypotonic solution has a lower solute concentration. When two solutions have the same osmotic concentration, the solution are isotonic. Equiparent facilitated osmosis. So this is a picture of hypertonic, isotonic, and hypotonic. Osmotic pressure. Osmotic pressure, force needed to stop osmotic flow. Cell in a hypotonic solution gains water, causing cells to swell, create pressure. If membrane store enough, cell reach counterbalance of osmotic pressure driving water in with hydrostatic pressure driving water out. Cell wall of prokaryotes, fungi, plants, protists. If membrane is not strong, it may burst. Animal cell must be in isotonic environment. Maintaining osmotic balance. Some cells use extrusion in which water is ejected through contravacuous isosmotic regulation involves keeping cells isotonic with their environment. Maria Icosonic adjust internal concentration to match the seawater. Tourist animal circulate isotonic fluid. Plant cell use turgor pressure to push the cell membrane against the cell wall and keep the cell rigid. Active transport. Active transportation required energy against concentration gradients. Uniport, symport, and antiport. Sodium and potassium pump couple transport. This is like three types of active transport. ATP is used directly or indirectly to fuel active transport. Move substance from low concentration to high concentration and require the use of highly selective carrier proteins. Active transport. Carrier protein used in active transport include uniporters, move one molecule at a time. Symporter move two molecules in the same direction, and antiporter move two molecules in opposite direction. Terms can also be used to describe facilitated diffusion carriers with no energy required. Sodium and potassium pump. Direct use of ATP for active transport. And its antiporter moves 3 sodium out of the cell and 2 potassium into the cell. Against their concentration gradients, ATP energy changes conformation of carrier proteins. This is a picture of a sodium and potassium pump. So you can see that it's antiporter. It's move sodium out of cell and put potassium in the cell. It's different direction. Couple transport. Use ATP indirectly. Use symporter. Molecules move against their concentration gradients using energy stored in gradients of different molecules. glucose sodium supporter capture the energy from sodium diffusion to move glucose against a concentration gradient. This is a picture of coupled transport. You can see that it's a supporter, so it move two molecules in the same direction. More sodium outside of cell. Sodium diffuse into cell coupled to glucose. Sodium potassium pump use ATP to pump sodium out of cell, create more sodium outside of cell. Glucose sodium supporter captures energy from sodium diffusion to move glucose against concentration gradients. Log transport. It has two types, endocytosis and exocytosis. Endocytosis. It's a movement of substances into the cell. Phagocytosis. Cell takes in particulate matter. Phagocytosis. Cell takes in only fluid. Receptor mediate endocytosis. Specific molecules are taken in after they bind to a receptor formation of chitin-coated pits and vesicle. This is a picture of endocytosis. You can see that it's move substances into cell. And exocytosis, movement of substances out of cell, required energy, discharge of material out of the cell, used in plants to export cell wall material, used in animal to secrete hormones, neurotransmitter, and a distinctive enzyme. So, in conclusion, passive transportation do not require ATP, and it go from high concentration to low concentration. It have three types: first, diffusion; second, facilitated diffusion; and third, osmosis. Active transportation is required in ATP. It's go low concentration to high concentration. It have three types, or it also have three types. First, uniport, symport, antiport. Second, sodium potassium pump, and third, coupled transport. Chapter 6, Energy Metabolism 1. Flow of energy, thermodynamic. Branch of chemistry concerned with energy changes. Study of energy flow of transformation. Kinetic energy to potential energy and potential energy to kinetic energy. Cells are governed by the law of physics and chemistry. What is energy? Energy is capacity to do work. First, kinetic. It's energy of motion. And potential, it's stored energy. Many forms: mechanical, heat, sound, electric current, light, or radioactivity. Heat, the most convenient way of measuring energy. 1 calorie equal to heat required to raise 1 gram of water 1 Celsius. When C is capital, equal to calorie, equal to calorie, equal to kilocalorie. How food energy measure 1000 calories 200 calories in soda is 2000 calories. Potential energy in the picture it's like its storage energy is based on her position. Also think of boulder on top of a hill. And kinetic energy, the energy in motion. She is able to slide down based on her previous position. Metabolism. Metabolism is total of chemical reaction carried out by an organism. Anabolic reaction or anabolism use energy to build up molecules. Anabolic steroid equal to build muscle. Catabolic reaction or catabolism release energy by breaking down molecules. Starvation break down muscle. The source of energy on Earth. Energy flow into the biological world from the sun. Photosynthesis. Organism, plants, capture this energy stored as potential energy in chemical bond in sugar made from photosynthesis. Redox reaction. Redox reaction is how energy is transferred at molecular level. Oxidation. Atoms or molecule loses an electron. Reduction. Atoms or molecule gains an electron. Higher level of energy than oxidized form. Redox. Oxidation and reduction reaction. Reaction always pair or weak. Oxidation is loss of electron. Reduction is gain of electron. Molecule A loses an electron, oxidized and loses energy. Molecule B gain an electron, reduce and gain energy. Loss of electron, oxidation or gain of electron, reduction, weak or weak. Oxidation is loss of electron, reduction is gain of electron. Laws of thermodynamic. First law of thermodynamic. Energy can't be created or destroyed. Energy can only change from one form to another. Total amount of energy in the universe remain constant. During each conversion, some energy is lost as heat. A second law of thermodynamic. Entropy is continuously increasing. Energy transformation proceeds spontaneously to convert matter from a more order less stable from a less order more stable form. Free energy. G is energy available to do work. G equal to H minus Ts. H is from entropy, that is energy in a molecule's chemical bonds. T is from absolute temperature. And S is entropy, it is available energy. G equal to H minus Ts. Positive G Positive delta G have more free energy than reactants, endergonic and anabolic require input of energy, not spontaneous. For example, photosynthesis. Negative delta G have less free energy than reactants. And exergonic catabolic release excess free energy, spontaneous. For example, cell respiration. This is a picture of positive delta G and negative delta G. Activation energy or EA. It is extra energy required to de-establish existing bonds and initiate a chemical reaction. Exergonic reactions rate depends on the activation energy required. The rate can be increased two ways. First, increasing energy of reacting molecules or heating. Second, lowering activation energy. Catalyzed substandard fluid inflow chemical bonds in a way that lower activation energy, makes reaction process faster, not use up in the process, cannot violate law of thermodynamics, cannot make an endergonic reaction spontaneous. Enzyme biological catalysts work by lowering activation energy. ATP cell energy. ATP is a short form from adenosine triphosphate. It's composed of a ribose, 5-carbon sugar, adenine, and chain of 3 phosphate. It's keys to energy storage, and bonds are unstable. ADP is 2 phosphate. AMP is 1 phosphate, and it's the lowest energy form. ATP cycle. ATP hydrolysis drive endergonic reaction. Couple reaction. The energy released by ATP is used immediately by cell for an energy requiring function. Result in net minus delta G. ATP not suitable for long-term energy storage. Fat and carbohydrate better. Cell storage only a few seconds worth of ATP. Energy from exergonic cellular reaction is used to regenerate ATP from ADP plus PI. ATP or its an energy created in mitochondria. ATP travels to where it is needed like the plasma membrane or sodium-potassium pump. Enzyme is biological catalyst, work by lowering activation energy. Most enzymes are proteins. Some are DNA core, RNA core, ribozyme. Shape of enzyme stabilize a temporary association between substrate Substrate is a substance on which the enzyme acts. Enzyme not change or consume in reaction. Enzyme active state. Active state, pocket or cleft of substrate binding, form enzyme substrate complex. Precise fit of substrate into active state. Apply stress to distort particular bonds to lower activation energy. Enzyme. The substrate sucrose consists of glucose and fructose bonded together. Then the substrate bind to the active state of the enzyme, forming an enzyme substrate complex. The bind of substrate and enzyme places stress on the glucose-fructose bond and the bonds break. Then product are released and the enzyme is free to bind other substrates. Enzyme function. Small molecules can affect function, coenzyme and cofactor. Rate of enzyme catalyzed reaction depends on concentration of substance and enzyme. Any chemicals or physical condition that affect the enzyme three-dimensional shape can change rate. Inhibitor substance that bind the enzyme and decreases its activity. Competitive inhibitor compete with substrate for active state. Non-competitive inhibitor bind to enzyme at a state other than active state. Causes shape change that makes enzyme unable to bind substrate. This is a picture of competitive and non-competitive inhibition. Allosteric enzyme. Allosteric enzyme. Enzyme exists in active and inactive form. Most competitive inhibitor bind to allosteric state, chemical on or off switch. Allosteric inhibitor bind to allosteric state and reduce enzyme activity. Allosteric activator bind to alloesteric state and increases enzyme activities. So the allosteric inhibitor and allosteric activator, it's different. The difference is allosteric inhibitor is reduce enzyme activity, but the allosteric activator is increases enzyme activities. Metabolism. Metabolism is a total of chemical reaction carried out by an organism. Anabolic reaction or anabolism use energy to build up molecules. Catabolic reaction or catabolism release energy by breaking down molecules. Biochemical pathway. Create the chemical reaction that create, store or produce other chemicals product for daily function. Reaction occurs in a sequence. Products of one reaction is the substrate for the next reaction. This is a basic metabolic pathway chart. Feedback inhibition. End product of pathway bind is an allosteric state on enzyme that catalyzes first reaction in pathway, shuts down pathway so raw materials and energy are not wasted. The first one is no feedback, and the second one is negative feedback. Most of our regulation are negative feedback. Chapter 7 Energy and Metabolism 2. Energy transformation. How organisms obtain energy. Autotroph. It produces their own organic molecules through photosynthesis, such as plants, and hetero, hetero, heterotroph. It eat organic compounds produced by other organisms such as animals. Cellular aerobic respiration. It's glucose and oxygen and it changed to carbon dioxide, water, and ATP. Delta G is minus 686 calorie mole of glucose. Breaking down sugar is endogonic. This is large amount of energy must be released in small steps rather than all at once. Cellular respiration is a series of redox reaction, many small steps, slow release of energy. Redox are coupled reaction. One molecule loses electron and one molecule gains electron. Dehydrogenation. Lost electrons are accompanied by protons. A hydrogen atom is lost one electron and one proton. To follow the electron, follow the H. Redox. The electron carrier. Redox reaction. Electron carry energy from one molecule to another. Electron carrier in cell respiration are NAD+ and FAD. So, reduced form. Sugar and food molecule has electrons to lose. Then it's oxidized sugar and it changed to oxidation. Sugar and food molecule lost electron. Oxidized form NAD+, ready to accept electron, and it's reduced NAD+, it will change to reduced form NADH, ready to accept electron. Respiration versus fermentation. Aerobic respiration is a final electron acceptor is oxygen, and anaerobic respiration final electron acceptor is an inorganic molecules or not oxygen. Fermentation final electron acceptor is an organic molecule converted into lactic acid or ethanol plus CO2. Cells use ATP to drive endergonic reaction. There are two mechanisms of synthesis of ATP. First, substrate level phosphorylation transfer phosphate group directly to ADP. Use an enzyme during glycolysis and Krebs cycle. Oxidative phosphorylation ATP synthesis uses energy from a proton gradient during ETC and chemiomosis. And this is substrate level phosphorylation. Aerobic cellular respiration. Oxidation of glucose. Steps in aerobic respiration. First, glycolysis. Second, pyruvate oxidation. Third, Krebs cycle. And fourth, electron transport change and chemiomosis. It occurs in this order. Glycolysis and Krebs cycle create little ATP but create electron carrier that all meet up in the step 4. ETC, all electrons carrier meet here, lots of ATP create. So this is a picture of the step in aerobic respiration. It's you can see that this is glycolysis, pyruvate oxidation, Krebs cycle, and electron transport change and chemiomosis. So let's see the first step, glycolysis. It occurs in the cytoplasm, convert one glucose to 2 pyruvate. 10-step biochemical pathway. Energy investment stage. 2 ATP are used to add to glucose. Energy produce stage. 4 ATP produced. Substrate level phosphorylation to NADH redox. NAD plus is reduced to NADH. NAD plus is gain an electron. 2 molecules of pyruvate. Pyruvate still have energy to be extracted. So this is a picture of glycolysis. You can see that it's used 2 ATP and then it changed NADH to NAD plus to NADH, and the result it will have pyruvate acid, 2 pyruvate acid. So step glycolysis. Location in cell is cytoplasm. Starting material is glucose and NAD plus, and end product is 2 pyruvate, 2 ATPs, and 2 NADH. This is free energy during glycolysis, pyruvate oxidation, and Krebs cycle. and there is only so much net plus present in a cell. There, we have to recycle recycle NADH to create more NAD+ to keep glycolysis running. NADH must be recycled for glycolysis to continue. NADH must be recycled to NAD+ by either aerobic respiration and fermentation. Fate of pyruvate. Oxygen present. Pyruvate is oxidized to acetyl coenzyme and its aerobic respiration. Oxygen no present. Pyruvate is reduced in order to regenerate NAD+ from NADH. Step 2. Pyruvate oxidation. It occurs in the mitochondria in eukaryote and occurs at the plasma membrane in prokaryote. Each pyruvate is oxidized, lose electrons that are collected by NAD+. So this is like conclusion. Located in mitochondria matrix of eukaryote, cytoplasm of prokaryote. 2 pyruvate starting material and end with 2 acetyl coenzyme, 2 NADH and 2 CO2. Step 3. Krebs cycle. Energy remains in bond of acetyl coenzyme. Krebs cycle transfer much of this energy to coenzyme NAD+ and FAD. Occurs in cytoplasm of prokaryote and in matrix of mitochondria matrix of eukaryote. This is picture of Krebs cycle. Krebs cycle. Acetyl group are oxidized to produce more electron carrier. NAD+ plus electron its NADH and FAD plus electron its FADH2. Biochemical pathway of steps in 3 segments. Each step is catalyzed by a specific enzyme. For each acetyl coenzyme entering, release 2 molecules of CO2, reduce 3 NAD+ to 3 NADH, reduce 1 FAD to FADH2, produce ATP, regenerate oxylocated so this is like a result. Electron transport change and chemiomyo-osmosis or step 4. Its location, it's in inner mitochondria membrane in eukaryote, plasma membrane in prokaryote. Electron carrier from previous step drop off the electron used to pump H plus out of cell and driven by oxygen. ETC is a series of membrane bonds, electron carrier embedded in the inner mitochondria membrane. Electrons from NADH and FADH2 are transferred to complex of ETC. Each complex a proton pump, creating proton guardians. Transfer electron to next carrier. Electron ends up at oxygen. Step ETC electron carrier of glycolysis. Pyruvate oxidation and Krebs cycle dropped off electron at ETC membrane complex. H plus is pumped out, create a gradient. Chemiomyosis. H plus fall back into matrix through ATP synthesis. ATP created. Oxidation without O2. Aerobic respiration is final electron acceptor is oxygen and anaerobic respiration final electron acceptor is inorganic molecules or not O2. And fermentation, final electron acceptor is an organic molecules convert into lactic acid and ethanol plus CO2. Aerobic respiration, methanogens. CO2 is used instead of O2. CO2 is reduced to CH+, found in diverse organisms including cows, sulfur bacteria. Inorganic substrate is reduced to hydrogen sulfate, H2S. Early sulfate reducer set the stage for evolution for photosynthesis. Fermentation reduce organic molecules in order to regenerate NAD+ for glycolysis. Lactic acid fermentation occurs in animal cells, especially muscle, such as during splitting. Electrons are transferred from NADH to pyruvate to lactic acid. Alcohol fermentation, no oxygen present, so NADH produced in glycolysis has no ETC to drop off electron and become NAD+ again. Need way to regenerate NAD+ to keep at least glycolysis going. NADH drop electron off at acetyl and becomes NAD+ again. Catabolism of proteins. Amino acid undergo deamination to remove the amino acid. Remainder group of amino acid is converted to a molecules that enter glycolysis or the Krebs cycle. Electron carrier create that go to etc chemiomosis for ATP production. Catabolism of fats. Fats are broken down to fatty acid and glycerol. Fatty acids are converted to acetyl group by B oxidation. Acetyl group join with coenzyme fatty acid yields 20% more energy than 6 carbon glucose. The key role of cell division, the ability of organisms to reproduce best, best distinguishing living things from non-living matter. In unicellular organism, division of one cell reproduces the entire organism. Cell division is an integral part of cell cycle. The life of a cell from formation is its own division. Cell division results in genetically identical daughter cell. Most cell division results in the daughter cell with identical genetic information DNA. A special type at division produces non-identical daughter cell, gamete or sperm and egg cell. Cellular organization of the genetic material. DNA molecules in a cell are packed into chromosome. Every eukaryotic species has a characteristic number of chromosomes in each cell nucleus. Somatic cells have two sets of chromosomes. Gametes have half as many chromosomes as somatic cells. Each duplicated chromosome has two sister chromatids, which separate during cell division. The centromere is narrow west from the duplicated chromosome, where the two chromatids are most closely attached. Eukaryotic cell division consists of mitosis and cytokinase. The cell cycle consists of mitotic phase and interphase. The mitotic spindle, an apparatus of microtubules that control chromosome movement during mitosis. Mitosis can divide into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. The cell cycle control system, regulated by both internal and external control. The clock has specific checkpoint where the cell cycle stops until a go-ahead signal is received. A binary fission prokaryote produced by a type of cell division, cyclin and cyclin-dependent kinase. Two types of regulatory proteins are involved in cell cycle control. MPF, a cyclo-CDK complex that triggers a cell passage past the G2 checkpoint into M-phase. Cancer cells don't respond normally at the body control mechanism, may not free growth factor to grow up, divide. A normal cell is converted to a cancer cell by a process called transformation. Malignant matter invade surrounding tissues and can metastasize, exporting cancer cells to other parts of the body. Variation on a theme. Genetic. The scientific study of heredity and variation. Heredity, the transmission of traits from one generation to the next. Variation, demonstrated by the difference in appearance that shows from parents and siblings. Genes, the unit of heredity, and are made up of segments of DNA. Asexual and sexual reproduction. In asexual reproduction, one parent produces genetically identical offspring by osmosis. In sexual reproduction, two parents give rise to offspring that have unique combination of genes from two parents. The sex chromosome are X and Y. Female have XX, male have XY. Autosome. There are 22 pairs that don't determine sex. Each pair of homologous from each parent. A diploid cell has two sets of chromosomes. For humans, the diploid number is 46. A gamete contains a single set of chromosomes is haploid. For humans, the haploid number is 23. In an ovum, the sex chromosome is X. The sperm cell, the sex chromosome may be X or Y. Fertilization and meiosis alternate in sexual life cycle. A life cycle, the generation to generation sequence of stage. Human somatic cells have 33 pairs of chromosomes. A karyotype, an ordered display of the pairs of chromosomes from a cell. Homologous chromosome, two chromosomes in each pair. Fertilization is an union of gametes. The fertilization egg is zygote. Gametes are the only types of human cell produced by meiosis rather than mitosis. Meiosis results in a set of chromosomes in each gamete. The variety of sexual life cycle. The alternation of meiosis and fertilization is common to all organisms that reproduce sexually. In animal, meiosis produces gametes. Gametes are the only haploid cell in animal, plants, and some organs exhibit an alternation of generation. The stage of meiosis. The first cell division, homologous chromosome separate. In the second cell division, sister chromatids separate. The sister chromatids are genetically identical and join at the centrosome. Prophet 1 chromosome begin to condense in synapse. Homologous chromosome loosely paired up. In crossing over, non-sister chromatids exchange DNA. Metaphase 1 telomere line up at metaphase plate. Microtubule from one pole are attached to the kinetochore. Anaphase pair of homologous chromosomes separate. One chromosome move toward each pole. Sister chromatids remain attached at the centromere. Telophase. Each half of the cell has a haploid set of chromosomes. Cytokinase usually occurs simultaneously. Prophase 2. A spindle apparatus form. Chromosome move toward to metaphase plate. Metaphase 2. The sister chromatids are arranged at the metaphase plate. The kinetochore of sister chromatids attached to microtubule extending from opposite pole. Anaphase 2. The sister chromatids separate. The sister chromatid of each chromosome now move as to every individual chromosome toward opposite pole. Telophase 2, the chromosome arrive at opposite pole. Cytokinase separated the cytoplasm. Each daughter cell is genetically distinct from the other and from the parent cell. Genetic variation produced in sexual life cycle contribute to evolution. Mutation are the original source of genetic diversity. Reshuffling of during sexual reproduction produce three mechanisms contribute to genetic chromosome. First, independent assortment of chromosome, homologous pair of chromosome randomly at metaphase 1 or meiosis. The number of domination possible when chromosome assort independently into gametes is 2n. Crossing over product recombinant chromosome. Crossing over begins very early in prophase 1. In crossing over homologous version of two non-sister chromatid plate. Crossing over contribute to genetic variation. And the last one, random fertilization. Random fertilization adds genetic variation because any sperm can fuse with any ovum. The fusion of two gametes produce a zygote with any of about 70 trillion diploid combination. Crossing over adds even more variation. Natural selection results in the accumulation of genetic variation favored